So what happens when we eat an egg? So unless we are eating it raw, the first step in egg is the digestion, right? So it involves chewing. So you just imagine what is happening when you eat an egg. Okay, so the teeth, what does it do? It will be masticating the egg into smaller pieces, and it will be directly swallowed because the salivary glands provide saliva to enable the process of swallowing, and will be there the partially mashed egg passes into the stomach through the esophagus. And what happens in the stomach, the gastric juice containing the HCl starts converting the pepsinogen into pepsin. Then only the digestion will be started or initiated. And this again, HCl also causes the denaturation. Of course, we are having the heat denatured egg. So when we prepare the egg, it is prepared under heat. So heat itself is causing denaturation. അതുകൊണ്ടാണല്ലോ എഗ്ഗിന്റെ കൺസിസ്റ്റൻസി അങ്ങ് മാറിപ്പോയി അല്ലേ? You can see it directly with your naked eyes. The denaturation is happening, and this denatured egg is further processed by the HCl also, enabling the digestion of the egg to happen in a more rapid manner. So the unfolding of the proteins, what do you mean by denaturation? The three-dimensional structure is lost. Okay. So the quaternary, tertiary, and the secondary level of organization of the proteins will be lost. Only the primary level will be remaining intact. So such a protein is known as a denatured protein. And this primary structure has to be now broken up. That is what is happening during the digestion. It is not that easy because they are made up of peptide bonds. We know that secondary level, tertiary level, and quaternary level of organization of proteins are made up of non-covalent interactions. So it is very easy to break such non-covalent interactions, but it's not that case in which the covalent bond. Okay, so the three-dimensional structure of the egg is lost, and the pepsin further dismantles the protein chains into smallest fragments, and they are again the globular proteins are now broken into the denatured form, and they are taken further into the intestine, where the small intestine, where the chyme will be acted upon by the pancreatic juice. The pancreatic digestive juice again contains trypsin, chymotrypsin, and carboxypeptidases. They causes the further digestion of the egg. And finally, the smallest forms, the ones the amino acids are formed, or the small tripeptides and dipeptides, that move into the intestinal lumen, where the intestinal juice, what does it do? Within the enterocyte, the intestinal juice further breaks up the intestinal juice is made up of exopeptidases. So you know the dipeptide means it is made up of dipeptide, which means it is having only two amino acids joined together by a peptide linkage. So it is an exopeptidase, right? So exopeptidase come and cleave the peptide bond, and it is broken up into two individual amino acids. So that's what's happening in the case of intestinal juice, or that's what's happening in the intestinal lumen or within the enterocytes, so that they are converted into their simplest form, that is a individual amino acids. Now what happens to these amino acids? The amino acids, with the help of transport proteins, there are various transport mechanisms. In the case of glucose also, glucose and galactose are carried out are entering into the blood through active transport, like with the help of ATP and carrier protein. They are taken into the blood. So similarly, the amino acids also will be taken into the blood with the help of transport proteins, right? Transporters, various transport mechanisms are there. There is no need of input of energy, but the entry of the amino acid from the enterocyte into the blood is carried out only with the help of carrier proteins. So it is a facilitated transport. So with the help of facilitated mechanism, these micronutrients are now entering into the blood, and they finally reach the liver, right? And what is happening in the liver? What happens? These amino acids will be further catabolized. If they are required for the body, again they will be subject for synthesis of proteins. Otherwise, they will be subject for catabolism, and the breakdown of, or the destruction of the amino acids results in the production of a compound known as urea, NPN. Right? It's a non-protein nitrogenous substance. Urea is not a protein, but it is having nitrogen in it. It is the end product of amino acid metabolism. Amino acids in the destruction that is resulting in the formation of, കാരണം amino acid, we know that it's having an amino group and an carboxyl group. Carboxyl group ബാക്കി എല്ലാം യൂസ് അപ്പ് ചെയ്ത് പോകും. അതേസമയം what happens to the amino group? Amino group will be released. So amino എന്ന് പറയുമ്പോൾ തന്നെ അറിയാം, it will be converted into ammonia. right? So amino group ആയിട്ടല്ല excrete ആവുക, it is in the form of ammonia. പക്ഷെ again there is a irony, that is ammonia is highly toxic to the body. ഒരിക്കലും body cells exposed ആവാൻ പാടില്ലാത്ത ഒരു compound ആണ് ammonia. So this ammonia നെ എങ്ങനെ എന്ത് ചെയ്യണം? It has to be first converted into the detoxified form. Ammonia is highly toxic. അപ്പോൾ ഈ toxic ആയിട്ടുള്ള ammonia നെ നമ്മൾ എന്ത് ചെയ്യും? It will be converted into a non-toxic form, and the non-toxic form of ammonia is known as urea. So urea is the, or urea cycle or urea formation that is a conversion of the ammonia to the non-toxic form. That is a formation of urea, right? It is a catabolic pathway of amino acids. So urea formation is nothing but the destruction or the elimination of ammonia, right? And from where does ammonia comes? Ammonia comes from the amino group of the amino acids. Clear? So it is the elimination of ammonia. It is a urea cycle. So before entering into the synthetic pathway, first we'll start with the catabolism. Okay, then we enters into the synthesis of various non-essential amino acids. Non-essential amino acids in the synthesis മാത്രമേ നമുക്ക് പഠിക്കേണ്ടതുള്ളൂ, because body dietary proteins അല്ലെങ്കിൽ dietary amino acids will be directly utilized by the body. അതേസമയം there are non-essential amino acids, that is it should be synthesized by the body. അപ്പോൾ synthetic pathway എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ ആകെ വളരെ കുറച്ച് amino acids വരുന്നുള്ളൂ, non-essential amino acids in the synthesis. Okay, so before entering into the synthetic pathway, we will first deal with the breakdown or catabolism of proteins. Okay, so because ammonia is toxic, the liver transforms it into urea, which is then transported into the kidney and is excreted in the urine. Urea is a molecule that contains 2 nitrogen atoms and is highly soluble in water. Our kidney function proper, or the kidney proper functions blood urea level. So the blood urea level or serum urea level is normally 20 to 40 milligrams per deciliter. If the value of urea is high, then it is indicative of what? Kidney failure. Kidney failure ആണ് indication. കാരണം urea form ചെയ്യുന്നതിനനുസരിച്ച് it should be excreted through urine, and we know that urine is formed in the kidneys. So if kidneys are not functioning properly, it is indicative of the blood urea level. If the blood urea level is elevated, is found to be elevated, if it's going beyond 40 milligram percentage, 40 milligram per deciliter, it's indicative of kidney dysfunction. Okay, so it's a good choice for transporting excess nitrogen out of the body, and amino acids are the building blocks that the body reserves in order to synthesize other proteins. More than 90% of the protein that taken into the body through the diet is not get broken down further into the amino acid monomers. Instead, they will be recycled and utilized for the protein biosynthesis. Like that of normal plastics reuse, amino acids will be recycled in the body, the liver, to synthesize proteins. And every day over 250 grams of proteins are dismantled. Dismantled means destructed or broken up into peptides. And they amount synthesis. So that's how the homeostasis is maintained. So 250 grams breakdown 250 grams and synthesize. Like that of the cholesterol also, right? Cholesterol normal body total or normal adult in a day will be synthesizing around 1 gram. 500 micrograms 500 milligrams come from the diet, and 500 milligrams will be synthesized by the body. Adhe 500 അല്ലെങ്കിൽ അതേ 1 gram തന്നെ നമ്മൾ ചെയ്യും, we will be excreting also. That's how the homeostasis is maintained. So similarly, proteins also, around 250 grams of proteins are broken up in a daily manner, and the same amount will be synthesized also. Okay, so if the rate of breakdown is more and the rate of synthesis is low, then again it is known as negative nitrogen balance. We call it as negative nitrogen balance. അതായത് ബോഡിയുടെ പ്രോട്ടീനെ ബ്രേക്ക് ഡൗൺ ചെയ്യുന്നുണ്ട്, പക്ഷെ സിന്തസിസ് നടക്കണില്ല, അത്ര തന്നെ സിന്തസിസ് നടക്കണില്ല. ഈ ബാലൻസ് അവിടെ 250നെ ബ്രേക്ക് ഡൗൺ ചെയ്യുന്നുണ്ട്, പക്ഷെ അതേ 250 ഉണ്ടാക്കാൻ പറ്റണില്ലെങ്കിൽ അതിനെ നമ്മൾ വിളിക്കുന്ന പേരാണ് നെഗറ്റീവ് നൈട്രജൻ ബാലൻസ്. അതേസമയം എന്താണ് ഇപ്പൊ അതാണ് സംഭവിക്കുന്നത് ക്യാൻസർ അല്ലെങ്കിൽ ഏതെങ്കിലും ഡിസീസ്ഡ് കണ്ടീഷനിലൊക്കെ ബ്രേക്ക് ഡൗൺ ചിലപ്പോൾ നടക്കും, പക്ഷെ സിന്തസിസ് അവിടെ നടക്കില്ല. ലിവർ ഇസ് ദ സിന്തറ്റിക് സൈറ്റ് ഓഫ് ഓൾ ദ ബയോമോളിക്യൂൾസ് അല്ലെ. അപ്പൊ ഇഫ് ലിവർ ഇസ് നോട്ട് പ്രോപ്പർലി ഫംഗ്ഷനിങ്, വാട്ട് ഹാപ്പൻസ്? ബ്രേക്ക് ഡൗൺ ഓഫ് കോഴ്സ് വിൽ ബി ഹാപ്പനിങ്, ബട്ട് സിന്തറ്റിക് റേറ്റ് വിൽ ബി റെഡ്യൂസ്ഡ്. അല്ലെങ്കിൽ ഏതെങ്കിലും ഒരു ക്ലിനിക്കൽ കണ്ടീഷനിൽ ക്യാൻസർ അങ്ങനെയുള്ള കണ്ടീഷൻസിലൊക്കെ എന്താ ഉണ്ടാവുക, നെഗറ്റീവ് നൈട്രജൻ ബാലൻസ് ഉണ്ടാവും. അതായത് സിന്തസിസ് നടക്കില്ല, അല്ലെങ്കിൽ ഇറ്റ് വിൽ ബി ഡിമിനിഷ്ഡ് വൈൽ ബ്രേക്ക് ഡൗൺ വിൽ ബി ഇൻക്രീസ്. സോ ദാറ്റ് കണ്ടീഷൻ വി കോൾഡ് ആസ് നെഗറ്റീവ് നൈട്രജൻ ബാലൻസ്. ഇതേപോലെ തന്നെ പോസിറ്റീവ് നൈട്രജൻ ബാലൻസ് ഇസ് ഓൾസോ ദേ. അവിടെ എന്താ ഉണ്ടാവുക, കാറ്റബോളിസം കുറയും, ആനബോളിസം കൂടും. അത് എപ്പോഴാണ് ഉണ്ടാവുക, ഫിസിയോളജിക്കൽ കണ്ടീഷൻസിലുണ്ടാവും. ദാറ്റ് ഇസ് വാട്ട്സ് ഹാപ്പനിങ് ഡ്യൂറിങ് ഗ്രോത്ത്. ഫസ്റ്റ് നൈട്രജൻ ബാലൻസ് കാണുന്നത് ആൾക്കാർ കുട്ടികളിലാണ് സാധാരണ കാണുക. പ്യൂബേർട്ടി ഒക്കെ എത്തുമ്പോൾ നമുക്കറിയാലോ, ഒരു 5-10 years ആയി കഴിഞ്ഞാൽ പിന്നെ പെട്ടെന്ന് ഒരു ഗ്രോത്ത് ആണ് കുട്ടികൾക്ക് ഉണ്ടാവുക, റൈറ്റ്? സോ ദാറ്റ്സ് ബിക്കോസ് ദേ ആർ ഹാവിംഗ് എ പോസിറ്റീവ് നൈട്രജൻ ബാലൻസ്. ദാറ്റ് ഇസ് ബ്രേക്ക് ദ റേറ്റ് ഓഫ് കാറ്റബോളിസം വിൽ ബി reduced while anabolic rate will be increased. So that is how പോസിറ്റീവ് നൈട്രജൻ ബാലൻസസ്. പ്രഗ്നൻസി, ലാക്ടേഷൻ, അവിടെയൊക്കെ അല്ലെങ്കിൽ ഒരു ട്രോമ, ഒരു സർജറി ഒക്കെ കഴിഞ്ഞ് കഴിഞ്ഞാൽ എന്ത് ചെയ്യണം, ടിഷ്യൂ റിപ്പയർ അവിടെ നടക്കും, അല്ലെ? അപ്പൊ ടിഷ്യൂയുടെ പുതിയ സിന്തസിസ് നടക്കും. ടിഷ്യൂ സിന്തസിസ് നടക്കാൻ എന്താ അർത്ഥം, സ്ട്രക്ചറൽ പ്രോട്ടീൻസിനൊക്കെ പിന്നീട് അവിടെ ആദ്യം ഉണ്ടാക്കണം. അപ്പോഴൊക്കെ അവിടെ എന്താണ്, പോസിറ്റീവ് നൈട്രജൻ ബാലൻസ് ആണ് അവിടെ ഉണ്ടാവുന്നത്, റൈറ്റ്? നൗ ഇഫ് ദ കാറ്റബോളിസം, rate of catabolism is equal to rate of anabolism, then we call it as nitrogen balance. That is, the balance is maintained. Okay. So to form the new proteins, amino acids from the food and protein destructions are placed into a pool. Actually, ഞാൻ പറഞ്ഞു amino acid pool എന്നുള്ളൊരു പൂളിനെ നമുക്ക് ഒരിക്കലും ഇവിടെയാണ് amino acid pool എന്ന് പറഞ്ഞാൽ നമുക്ക് ഒരിക്കലും കാണിക്കാൻ പറ്റില്ല. പക്ഷെ there is a theory that there is an assumption that ബോഡിക്ക് എവിടെയോ ഒരു amino acid pool ഉണ്ട്, അവിടെ പോയിട്ടാണ് നമ്മൾ ആവശ്യമുള്ള amino acidsനെ പിക്ക് ചെയ്യണത്, സിന്തസിസ് നടത്തുന്നത്. സോ എവിടുന്നാണ് ഈ അമിനോ ആസിഡ് പൂളിൽ അമിനോ ആസിഡ്സിനെ കിട്ടുന്നത്? ഇറ്റ് ഈസ് ത്രൂ ഡയറ്ററി പ്രോട്ടീൻസ്, അല്ലെങ്കിൽ ഈ പറഞ്ഞ പോലെ പ്രോട്ടീൻസിന്റെ ബോഡിയിലെ ടിഷ്യൂ, ഏതെങ്കിലും ടിഷ്യൂ പ്രോട്ടീൻസ് ഒക്കെ കാറ്റബോളിസിലേക്ക് പോകുമ്പോൾ that proteins will be undergoing catabolism and these amino acids will be channeled into this amino acid pool, right? So that though it is not a literal pool, when an amino acid is required to build another protein, can be acquired from the additional amino acid that exists within the body. So it is critical to maintain the amino acid levels within the cellular pool by consuming high quality proteins in the diet. അതുകൊണ്ടാണ് protein rich ആയിട്ടുള്ള ഫുഡ് കഴിക്കാൻ വേണ്ടി പറയുന്നത്. So that amino acid pool is rich in all the amino acids. So whenever the demand arises, the body can directly go and pick up the required amino acids. So proteins, they are the primary constituents of the body. A regular and adequate supply of protein in the diet is essential for the integrity and function. Dietary proteins are the primary source of nitrogen, and adult man require around 70 to 100 grams of proteins per day. And they have got different functions, that is, they are utilized for body synthesis of body proteins. Carbon skeletons of amino acids are used for energy production. Then carbon and nitrogen atoms are used for synthesizing other nitrogenous and non-nitrogenous substances. Okay, so how does the digestion happen? Digestion is a disintegration of complex nutrients into soluble and assimilable form, that is simplest form. Proteins are too large to be absorbed. So what is happening? The dietary proteins are hydrolyzed into amino acids with the help of proteolytic enzymes, which can be easily absorbed. They are responsible for degrading the proteins, and they are produced by different organs, the stomach, pancreas, and the small intestine. So this is what is happening. The amino acids A1, this is one first amino acid, second, three. So this is a pentapeptide. Okay, pentameric molecule. So this pentameric molecule is having they are joined together by 4 peptide linkages. So these peptide linkages are broken up. This is known as a primary structure. Okay, the linear arrangement of amino acids. So by means of the covalent bond, the peptide bond. So during the digestion, what is happening? These peptide bonds are undergoing breakdown. So proteases will break the peptide bonds into individual amino acids. So peptidases are of 2 types, endopeptidases and exopeptidases. Endopeptidases are pepsin is a typical example, while exopeptidases the example are aminopeptidase and carboxypeptidase. You know that we will be having, if it's a pentameric molecule, it's having a carboxy terminal end and amino terminal end. By e amino acid, it's having an amino group. This amino group, carboxy group joins in the peptide bond. You know that peptide bond is CONH bond. Okay, so this is the N-terminal end and this is the carboxy terminal end. About exopeptidases and exopeptidases, they act only on the terminal regions. So one terminal position carboxy terminal we call it as enzyme we call it as carboxy peptidase. This is aminopeptidase. So it can act only on this aminopeptidases can act on the N-terminal end, while carboxypeptidases can act only on the C-terminal end. Okay, so such an enzyme is known as exopeptidase. That is, if an enzyme is coming and acting in this particular peptide bond, it's an endopeptidase. Okay, so again the peptidases, they are released in the zymogen form, which is known as the proenzymes of inactive form. So the digestion of proteins, stomach, hepatic juice, pancreatic juice, here the absorption happens and they will be taken into the liver.